In this video, we will talk about the human birth defects. We will go through the 4 types of them, their cause, and give each of them an example they'll discuss. Chromosomal abnormalities. It is a type of genetic defects of infant, often caused by abnormalities in parental meiosis or mitosis in the fetus, and it can be induced by other factors to cause mutation in chromosomes. The subtypes can be classified into 2 groups: the autosomal and sex chromosome origin, or numeric and structural division. And in numeric defects, there are trisomy and monosomy, while in the structural defects, there are translocations, deletions, microdeletion, ring, duplication, inversion, isochromosome. For example, we will discuss the Patau syndrome. It is caused by the trisomy of chromosome 13, and it comes in 2 types. Full trisomy, caused by non-disjunction during meiosis, and mosaic trisomy, caused by non-disjunction during mitosis. Study shows that, as other trisomy disease, it has higher incidence rate as the maternal pregnant age increase, and the incidence rate is somewhere between 1 in 10,000 to 2,000. The symptom often includes microcephaly, hollow prosencephaly, polydactyl, cleft lips, low-set ears, etc. Mutant genes. The cause of mutant genes are change in base pairs, which can be classified to point mutation, including transversion and transition, friendship mutation, including insertion and deletion. Note that all the gene mutation obeys Mendelian law as well. For example, we'll discuss achondroplasia. It is caused by the mutation in fibroblast growth factor receptor 3 gene, which interferes with how this protein interacts with the growth factor associated with bone production, like transformation of cartilage into bone. The effects of achondroplasia is genetically dominant, with 1 mutant copy of the FGFR3 gene being sufficient to cause the syndrome. Also, the frequency of mutation increases with paternal age, 1 in 1,875 for father over 50 years old compared to 1 in 27,500 in the general population. The symptom contains dwarfism, short proximal limb, macrocephaly, frontal bossing, varus and valgus. Environmental agent. As the name indicates, it is related to the environmental factors called teratogens. It includes radiations, virus like rubella, Zika, cytomegalovirus, drugs excessive or insufficient of particular substance like alcohol or folic acid, and mechanical factors like nucleocor. They mainly affect tissues in rapid differentiation phase while they are most susceptible in this stage. For example, we will discuss the warfarin. Original use of warfarin as an anticoagulant drug by blocking the vitamin K to utilize the coagulation. Due to its low molecular weight, it can trespass the placenta of the fetus, causing internal bleeding of the fetus and inhibition of osteocalcin by inhibition of vitamin K, which either cause abortion or stunt bone mineralization. The symptoms include low birth weight, slower growth, mental retardation, deep groove of the midline of the nose, deafness, microcephaly. Clinically, warfarin can be replaced by the heparin. Multifactorial inheritance. It is caused by the combination of genetic and environmental factors, which result in changes of multifactorial trait. For example, we will discuss the cleft lip and palate. Cleft lip and palate can both be divided into complete and incomplete type, based on the cleft depth and length as shown in the picture, and complete to complete from left to right. Also, compared to midline, it can classify further into unilateral and bilateral types. And it is also worth notice that, in cleft palatine, the split uvula is often observed. Genetic factors contribute 20 to 50% of this syndrome, while environmental factors contribute to the others. The genes involving the deformation include COPTP1, GAD1, HYAL2, IRF6, PVRL1, MSX1, PMP4, and PHF8. IRF6 and PHF8 especially cause van der Waals syndrome and the Siderius X-linked intellectual disability individually. The environmental factors include smoking, alcohol, and other chemical substances as shown. Smoking causes maternal hypoxia and leads to the mutations in the gene PHF8, while alcohol causes abnormal growth of the neurocrest cell.